Hi, welcome everybody. We'll take just a minute for a few more people to log on. Welcome. Awesome. All right, I think we'll go ahead and get started. Welcome everyone to Hawk Mountain Sanctuary Stay-at-Home Speaker Series. Today's program is Bat Community and Infection Change Across a Mosaic Landscape with Thalia Williamson. Hi, Thalia.
Hello.
My name is Riley. I'm an educator and raptor specialist at Hawk Mountain Sanctuary, and we're so glad that you are all joining us today. Just to share a little bit of information about Hawk Mountain before we begin. Hawk Mountain Sanctuary is the world's very first refuge for birds of prey, and we continue to work hard to be leaders in raptor conservation, science, and education locally and globally around the world. Hawk Mountain Sanctuary is a private nonprofit, and membership is the lifeblood of our organization. To all of our members, thank you so much for your continued support. And if you're joining us today and you're not a member, we hope that you consider becoming one in the future. Hawk Mountain hopes that everyone is remaining healthy and safe during these times of COVID challenges. And we're excited to offer our local and global community a variety of free virtual programming. And as always, Hawk Mountain greatly appreciates and depends on donations. Just so everyone is aware, today's program is being recorded. So the video will, after the video is finished, it will be available on our YouTube channel as a continued resource. If you'd like to go to our YouTube channel, you can head to our website, hawkmountain.org, to get that link. And at any time during today's program, viewers can submit questions through the Q&A feature. It's down at the bottom of your screen. It's a little bit different from a regular Zoom meeting, but you can hit Q&A, type in any question that you have, and we have some time put aside at the end to answer some of your questions. We are so excited that Thalia is joining us today to talk about her experiences and insights from her graduate fieldwork studying bat communities in Ecuador. Before we get started, I'm just going to share a little bit of information with the audience. So Thalia was actually a trainee at Hawk Mountain Sanctuary during the summer of 2019. And after that, she became a field technician for 2 months in Southern Australia, helping collect data on heat stress behavior and body condition of the Jacky winter bird. Currently, she is pursuing a 1-year MS at Tulane University in ecology and evolutionary biology. As part of her program, she has traveled to Ecuador in the summer of 2021 and collected data on bat biodiversity and pathogens across different land use types, which we'll be learning more about today. So Thalia, thanks so much for joining us. I'm going to put myself on mute, turn my camera off, but I'll still be here, but you have the floor. You can go ahead and share your screen.
Awesome, thank you. Okay. Can everyone see that? I think it's the correct one now. Yeah, okay. Hi everyone. Like I was introduced, I'm Thalia Williamson, and I'll be talking about bat community and infection change across a mosaic landscape. So the outline for our presentation. Yeah, our outline for our presentation. Sorry, just making sure that I'm sharing the correct screen. Is we're going to talk about background, methods, hypotheses, and some preliminary results, and then some future directions. So a little bit more about me. My personal background, I started at the University of Richmond, where I played field hockey and was on full scholarship. Unfortunately, I got injured, and I ended up going to Albright College as a part-time student. I never intended to stay at Albright. I really wanted to try and play field hockey again, but I had 2 really great advisors, and as well as like other professors who convinced me 2 days before the semester started to 2 more classes to my schedule so that I could become a full-time student. And so I stayed at Albright and I did an honors thesis looking at the impact of sound pollution on bird behavior. And I'd say this is probably where my scientific journey started. So after graduating in 2019, I immediately went to Hawk Mountain and I became a trainee. And there I got to spend a lot of time with American kestrels. And I also got to watch some bat extractions, which I had done while I was at Albright. And I also got to do butterfly counts, as well as I learned how to extract birds from mist nets. And then after that, I went and I participated in the Jacky Winter Project in Australia for about 2 months. And here is what the Jacky Winter bird looks like. It is most closely related to our American robin. The nest is on the bottom. And what I did there is I looked for nests, and then we also weighed birds and walked around following them to find them exhibiting heat stress behaviors. So in the crook of this branch, that is a Jacky Winter female. And what she's doing is she is kind of stretching out her wings and trying to shade the eggs because it is a very hot day and there's no shade. And after this, I was at Albright for another year where I worked in the student accessibility office. And then I ended up at Tulane. So that is where I'm currently. I'm a master's student at the Tulane University in Louisiana. And I worked with bats this past summer. So a little bit about the tropics and what I was doing. So in the tropics, there's a variety of reasons for deforestation that are occurring currently. A lot of it is agricultural expansion. So people are cutting down the forest for either planting crops like cacao or banana or palm oil, or they're cutting down the forest for pasture for cattle. They're also cutting down the forest for lumber production as well as increased infrastructure. And some of these effects can be quite disastrous. Desertification can occur where trees are cut down, and what ends up happening is the area dries out because there's no longer trees to keep the ground covered and shaded, and so you lose a lot of water in the area. And also with this, habitat loss for native species and endangered species occurs, which can have drastic consequences. And also there's something called spillover events, which is where disease can start transmitting more rapidly between organisms and humans because there's more interaction between the humans and the wild animals. So a little bit about bats. So why do we study bats? They provide great ecosystem services. Bats are great seed dispersers. So you have frutivores in the tropics, and they will eat the fruit, fly around, defecate, and deposit seeds in other locations. You also have pollinators. So there's quite a few nectarivore species in the tropics. And when they are getting the nectar from the flowers and moving from plant to plant, they're dispersing pollen and pollinating a lot of different species. And then they also contribute to pest control. And this is probably something that we can all really relate to and are more familiar with because we have a lot of insectivore species in the US. And so they do a great job of eating lots of insects and decreasing disease such as malaria. And they're also considered sentinel animals. So they're great bioindicators because certain species are specifically sensitive to different habitat changes in different regions. And so if we can understand what's going on and identify if the bats are impacted, we have a good idea of what's happening in the region. Now, a little bit about bats and disease. So bats are considered reservoir species. And that is because they obtain a lot of diseases like rabies and other things, but they don't actually express symptoms. So they have a very unique immune system. And one of the ideas is that flight causes a lot of oxidative damage. And so there's a increased metabolic demand of flight that leads to greater immunity. However, this needs a lot more research. And just because they can be reservoir species doesn't mean that they don't get sick. So one of the really good symptoms of bats getting sick is white nose syndrome, which is prevalent in the Northeast, but less so in the tropics. So a few questions that we had before we went down were, how do land use change influence community composition, sex ratios of species, parasite load, and disease prevalence? And hopefully we'll be able to answer a few of these questions, or at least give some hypotheses as to what will occur in the next few slides. So a little bit about our methods. So we were in the Mache Chindu Ecological Reserve in northwestern Ecuador, and we were at a field station called FCAT. And FCAT is unique because they are starting a reforestation project to see what occurs. They've been buying up land, a lot of agricultural land, and they're going to be replanting it to see how bat diversity changes across those different types of habitats over time. So what we were doing as one of our reasons for being down there was we were the start of a latitudinal study, which means they're going to be doing lots of biodiversity monitoring while every year they go down to see if there's any changes in bat species composition over time as reforested areas grow back up into secondary forest. And in this bottom corner, that's our station. Don't let anyone fool you. That is plastic tarp on the outside of our dining area. So this is just a diagram or a figure of all the different points that we could sample from. And so the samples that we took, we randomly chose sites based on accessibility, as well as trying to maintain as much distance from one another, so that there was to account for clumping and trying not to clump sites together. And so we sampled each site twice, about 3 weeks apart. And we did this through mist netting. Now, mist netting is also something that you can do for birds, but with bats, it's a little bit different. The bat bags, there's trammels in the mist net, and the bags that each of those trammels create is larger for bats, or larger for birds as opposed to bats. And so this is just some pictures of what we did. So first of all, to get to these sites, we had to actually either walk very, very far, or in some cases, we used a burro, and that's me on the burro after we dropped our stuff off at a site. And there were some nights where my Apple Watch was telling me that I climbed 100 flights of stairs, or we walked 18 miles that day. So field work is very intense, and it's not for everyone, but I really enjoy it. So here's also a picture of where our site is, and we had to climb down this steep incline to set up a station, and we then had to set up mist nets. And so this photo right here is us setting up mist nets. They're this fine mesh. Like I said, they have these trammels that go across, and they form these small bags. And so after we set up the mist nets, then we set up the processing station, which is right here. We're actually in the process of processing bats in this photo. But we would go out around 4:30, depending on where the site was, set up everything. Typically the nets were closed, although we did have to change our protocol a little bit. We had originally opened the nets at 6 to 11, but we caught a ton of birds. So we ended up having to change when we opened the nets. So we changed to opening the nets to 6:30. And here are some of the birds that we caught. We caught a ton of nightjars. That is this is that bird right here. I think we caught a total of like 6 or 7, which was a decent amount. We actually had one that flew right into, we were at a processing station and one bird flew right into my partner who was processing. It was kind of hysterical. But we're not talking about birds, we're talking about bats. So what do we do when we caught bats? So what ends up happening is you open up the nets and hopefully you get a bat to fall into them or fly into them. Now, the bats should be able to echolocate and kind of see the nets, but the idea is there's such a fine mesh that even if they identify that there's something there based on echolocation, they probably think it's either spider web or something, and so they think they can fly right through it, and they can't. So they then fall into the pocket. That's what's happening here, and someone has to go get them out. And so we wear protective gloves and we extract them from the net, and then they get put into a cloth bag. That's this bag right here. And so they're perfectly fine. You do this also with birds. You put them in a cloth bag to take them back to the processing station. And in this case, we hung them on a little tree branch. And so if we had multiple bats in a line, they'd hang on this tree branch and wait to be processed. And the bats our protocol said that we could have them for 3 hours. Most bats were not with us longer than, at the most, and on our busiest nights, no longer than an hour and a half. And then after you're done with whatever bat you're processing, you take this bag and you weigh it first, because you want to know how much the bat weighs. So this is one of my colleagues, this is Bill, and he is weighing the bat bag to get the initial mass, and then he'll weigh the bag after he's done processing the bat to figure out how much the bat actually weighed. And then after that, we take a couple of samples. So we take an oral swab that's shown here, and we're doing this to detect viral DNA or RNA. And then what we do is we take a blood sample. So that's what's happening here. And this is me taking a blood sample as well. This part was what I thought was going to be the worst part for me. I did not know if I could do this. If you know me, I hate needles. I think they're terrible. I definitely have a phobia of them, but surprisingly, it was one of my favorite things to do. And in some ways, I felt like it was retribution for when they bit me. So if they bit me, I was going to try and take some of their blood. And then after that, we would do a wing punch. And so this was to collect DNA. So this is, you use this little tool, a biopsy tool, and you just press down on the wing to get a little hole. And we took 2 of these. But what you do is you lay the wing flat so you can see where the blood vessels are, because if you do this properly, the bat should not bleed at all. And right here is the 2 holes, 2 wing punches that were taken, and it doesn't hurt them at all. Here is a bat later on in the season that has a healed. This was most likely a recapture, and so it's healed already within those 3 weeks. And then here's just another injury that we found, and it looks like it had broken one of its digits, and they fly with broken digits and they seem to do fine. So taking a biopsy doesn't really hurt the bat. And then what we do is we do parasite collection. So here's a bat that we captured, and here are some parasites. This part is very, very difficult. It is like playing Operation, but with moving parts. But once you get them, you use a tweezer, you grab the parasite, and then you put it in ethanol to kill it. And these guys are important because I'm using them for Bartonella detection. So Bartonella is a species of bacteria, and flies are an integral part of Bartonella transmission. And so this is a picture of a few different species of bat flies. And what you do is after you get the bat flies, and I've been working on some data from Costa Rica currently. And so what you do is you take these bat flies, you identify the species, and then you have to grind them up and extract the DNA. And then you have to run a PCR, and then you get a product, which is this gel right here. So you have to run a gel, and we have some Bartonella. So this yellow box is 2 definite bands of Bartonella. This one could potentially be a band of Bartonella. We're not sure. We're gonna have to retry and run that that bat fly. But that's the whole goal, is to find if there's Bartonella present or not. So our hypotheses are: 1, does biodiversity in forested sites is greater than agricultural sites? 2, species composition would vary according to landscape. And 3, sexual composition would vary according to landscape. So let's check out the results. So this is a phylogeny of all the species that we trapped this field season. We caught a total of 505 individual bats, which is a crazy number. And that represented 39 known species from 4 different families. Now, these boxes in the middle are representative of where they were caught. So the green boxes are forested sites, the red boxes are reforested sites, and the blue boxes are agricultural sites. And I will remind you about this throughout the presentation as we go on. And we will be focusing on the Phylostomidae family. And so first, we're going to talk about these red boxes. So these red boxes are 2 clades of nectar-feeding bats. And based on this, they seem to be more present in our agricultural sites. So they do better, and they seem to just be more active in agricultural sites. At least that's where we were catching them the most frequently. And this could be because in those agricultural sites, they could have lots of flowering trees and lots of plants that need to be pollinated. And so they just like those open areas a lot more. Next, we're going to talk about these gold areas. These gold boxes are insect feeding bats and also omnivores. And so they're mostly found in the forested site. There's one time that one of them was found in a reforested site, and none of them were found in agricultural sites. So this seems to mean that they are good bioindicators because they need really high quality habitat to be present. So without the forested sites, they would not be around. And then we have this yellow box. So this yellow box are our fruit-eating bats, and we find them pretty much everywhere. So they don't seem to care really what type of habitat they are in. They seem to just do very well in all different types of habitats. And this could be because there's fruiting trees both in the forested areas, the reforested areas, and the agricultural areas. And one of the species that we caught the most frequently was Corolla. And Corolla eat a plant called piper. And piper is a very shrubby, small plant. And it was very common in our reforested plots. So it could be that the piper just does well in disturbed areas. And that could be why we're finding the Corolla so frequently in all sorts of areas. Now, this is just a big picture graph, or not picture graph, a lot of it's a collage of bats that we caught this season. So I just want to point out a few. So this is a Thyroptera. They have these suction cups on their thumbs, and so they are bats that live or roost in rolled up leaves. And so they use those suction cups to kind of stick onto the sides of the leaf. This is a vampire bat. We caught two of these this summer. And what was interesting about these guys is the two that we caught had bald heads, which is unusual. And one of the hypotheses was that a lot of the farmers in Ecuador use a pesticide, not a pesticide, yeah, pesticide on their cows. And vampire bats are sanguivores, so they drink blood and they crawl up the cow's legs, and so they might be getting that pesticide on them and they're losing hair because of it. So that could be a potential question that one of my colleagues will be answering. And then we have a Tracops, which is a frog-eating bat. We caught one of these this season, and that was just very exciting. And then we have this guy, and that one's just funny because we found there was a photo in our bat book of this species, and our field assistant and field guide and the native Ecuadorian who was helping us, he saw this photo, and the bugs are just crazy when you're processing because you have a light on, and so the cicadas are pretty intense. And so he picked one up, and the bat had its mouth open, so he put the bug in its mouth, and the bat just sat there with the cicada in its mouth. It did not eat the cicada, but it was a pretty cute photo op. So we also did species accumulation curves. And so what we found is there was the greatest species richness in the forested areas, followed by reforested and then agricultural. So that wasn't too much of a surprise. But what's interesting is these curves have not plateaued yet. So that means that even though we sampled quite extensively and we caught 505 individuals and 39 species, there are probably still species in the area that we have not caught. So this is very exciting. And as part of that latitudinal study, this should change. But it was pretty interesting preliminary results. Now, these graphs are looking at the sites and how closely related the sites are. So this graph over here is just an example for comparison. This is not our actual data. This is the actual data that we collected this summer. So if the sites were not closely related, you would see something like this. So the sites are very interspersed with one another, whereas here, what you would hope to see is that, and what we do see, is that forested sites seem to cluster together, reforested sites seem to cluster together, and agricultural sites seem to cluster together. And so this is just showing that the types of sites, so forested, reforested, and agriculture, are more closely related to one another than they are to the other types of habitat. So this means that how we're categorizing the sites is a decent way to categorize them. And then this is just looking at the landscape and sex ratios and reproductive status. So we don't see any impact of landscape on reproductive status, and it seems to be relatively evenly distributed between males and females in the different sites. So what are our conclusions from this? So we can conclude that landscape can influence and could potentially predict bat diversity. Landscape type does not seem to impact reproductive status in bats. And so that's a good thing because we're not losing a significant proportion of one sex depending on the landscape. So what can we learn? So the effects on diversity are important because bats are important for seed dispersal, pollination, and also pest control. So if we're losing a lot of bats especially if we're losing a lot of insectivore bats in agricultural areas, and that's all that's surrounding a town, there could be impacts of greater disease, of like malaria and things like that from insects. Reproductive findings are important, and I kind of said this already, because if we're not seeing any differentiation between the sex ratio and reproductive status. That's a good thing. We don't want to see reproduction dropping really a lot in like forested areas or anything like that, because some species are more prone to wanting to be in certain areas. And so if reproduction is changing across landscape type, that could be problematic for species survival. So a little bit about our future work. We're planning on analyzing the wet samples that we collected. They are still in Ecuador. International field work can be difficult for that reason. So even though we did this last summer, we still have not gotten the samples from last summer. But so we're planning to analyze those samples that we collected and look at pathogen prevalence and like the patterns, and see if there's certain species of bats who are carrying disease more often than others. And then currently there's 2 PhD students in my lab, Hannah and Phoebe. They're both first year PhD students, and they are very excited to start their own projects this summer. And so Phoebe will be looking at cortisol in feces and looking at disease transmission through viral presence on flowers. So this is a nectarovore, and so she's seeing if nectarovores, if they go back to a flower that another infected individual was at, if there's the possibility of viral transmission. And then Hannah is looking at spillover potential. So she's looking at interactions of bats with domestic animals and people. So she's trying to see how many times people interact with bats and if there's the possibility of disease transmission due to that interaction. And so my future directions, I just recently accepted a PhD spot at Auburn University, and I'll be working with a colony of zebra finches. I have no idea exactly what my project will entail. The lab does a lot of heat stress work, but I am excited to continue my scientific journey and get a PhD. So I would like to acknowledge a few people. I'd like to acknowledge Tulane School of Science and Engineering, Dr. Hannah Frank, my PI, and the BAT team. And I would also like to thank Hawk Mountain for giving me this opportunity to speak about my research and to just do a little bit more outreach. So without further ado, does anyone have any questions?
Awesome. Thank you so much, Thalia. Yeah, if anyone has questions right now, feel free to add them into the Q&A section at the bottom of your screen, and we can go through and answer those questions. I actually wanted to kick off. I had a question for you. So I know you said that the research that you were doing was out in Ecuador, but I was curious if your experience from doing that research or just from being here in Pennsylvania for schooling, like if you have any information to share about like the local bat populations, like if there's any like crossover, like just if you could speak more about that.
Yeah, so I don't do a ton of work with bats in like the Northeast and in the United States. We in the US there are a little bit more like strict protocols with working with bats just because of white nose syndrome.
Mm-hmm.
And it's also just a lot harder to trap bats because a lot of them are insectivores, like I said, so they're much higher in the trees and like in the sky. So the majority of the bats that we caught this season in Ecuador were frutivores. So we do not catch a lot of insectivores just because they're so much higher in the canopy. So that's a big difference. Right. A lot of I think a lot of the bat work in the US is much more cave focused, because if you find a cave, you can put a net right outside so that when they fly out at dusk, you can catch them. But you're not necessarily going to be able to do a lot of just trapping in the open or like in a forested area. It's going to be much harder to actually catch anything.
Gotcha. And then my one other question I had for you was, do you think that people should be like concerned or like vigilant about, I know you were talking about how bats have a very unique like immune system. So the way they like have disease and pass that on, like, do you think that's something that people should have like a concern for?
That's a good question. Not necessarily. I mean, there is always the risk, like I said, they can have rabies. But at the same time, it's one of those things that if bats are behaving normally, if they're flying at night and things like that, they are probably doing normal bat behavior. So they most likely aren't. I mean, they could be sick and carrying something, but like it's not like if you see a bat during the day, don't touch it. Like we had a bat at one point fly into a net during our birders were misnetting during the day, and they had a bat fly into it.
Mhm.
And I couldn't believe one of our like head researchers, he went and got it out, and I could not believe he touched it because I was like, it's during the day, it's probably sick. But so if I mean, there's always cause for concern. Like I wouldn't say go into a cave and like lick the ground or something, but just be conscientious. I mean, raccoons, possums, other things carry disease too. You just need to be aware.
Right. All right. Well, I have a couple questions here from the audience that I'll go through. The first question is from Dr. Lori Goodrich, and she asked, congrats on the position. And she was wondering if Ecuador has the highest diversity of bats. She said that it seemed from like the photos and stuff that it looked very diverse.
I don't know if Ecuador has the highest diversity. I would a lot of the tropics is just much more diverse than the United States. They just have a higher concentration, and they just have a lot more resources, so species can differentiate. So they have like the insectivores, the frutivores, the sanguivores, and the nectarivores. So they're just able to find different niches a lot more than they can here. But I cannot speak to which country or South American country or even Australia. One of the tropical places has the most diversity. I just don't know which one.
Mhm. Awesome. My next question is from Patricia, and she asks if you had a favorite bat species that you worked with.
Oh gosh. Bats are fun. I prefer birds. But I will go back to the slide with all the bat photos. I would say I absolutely adored the Thyroptera. They're these guys, the suction cups. They're just so funny. They're very tiny. So they can bite you, but not nearly as much as some other ones. We weren't allowed to touch the vampire bats because they can bite through gloves. They can bite through raptor gloves. So they're a little crazy. So we weren't allowed to touch them. But I would have to say it's either the Thyroptera or this guy or the Trachops, just because they're so crazy. So these are the frog eaters. And so that was just so interesting to see. And compared to some of the other large bats, the the Trachops was very chill. Some of them are not very happy to be caught, and they're very bitey, and it's just not fun. But the Trachops was pretty nice and chill, which was exciting.
Very cool. All right. And my very last question I have here is from Jen, and she asked if there are differences in extraction between like extraction methods between bats and birds.
Yeah. So yes, there are. So bats, I would say, are probably a little easier than birds, just because the bats, with birds, their feathers get in the way. You cannot see where the mistnet is, which makes it really hard. For both, you kind of go in and grab their feet. But with birds, they grab onto the net with their feet and they don't let go. Whereas bats are more likely to be trying to like bite at the net. They try and chew through the net. So if you don't get them in time, they can be really tricky to get out because they'll bite through and get more tangled. But I think the different types of mist nets try and account for those differences in like extraction and like the way in which the organisms try and get out of the net and things like that.
Gotcha. All right. Well, thank you everyone for the really good questions. And thank you, Thalia, for such an interesting presentation. I learned more about bats today. So thank you all so much. I did want to plug an upcoming program that we are doing at Hawk Mountain Sanctuary about bats. It's May 21st. We have a local organization called the PA Bat Rescue, where they will be doing a live bat release, and you can learn from our stewardship team how to create a habitat for a bat and take it home with you. So if you're interested, definitely check that out on our website. Once again, Thalia, thank you so much. And thanks everyone for joining us. And yeah, hope everyone has a great night. Thank you. Bye, everybody.